Good morning, good afternoon, good evening to all those who have registered and participated in this webinar. I'm making this introduction on behalf also of Peter Holby, who is the director of the PSI, the Pandemic Science Institute at the University of Oxford. And the PSI is a new multidisciplinary institute within the University of Oxford, who's covering basic science, clinical science, including vaccine and drug development, social sciences, with a focus on translational research to deliver practical interventions to reduce the burden of and the risk of epidemic and pandemics. This webinar, the overall objective of this webinar is about the fact that I believe we kind of all agree that we need, in order to respond and to address outbreaks when they and where they occur, we need a rapid adaptation of technology platforms in order to respond, but also to prevent pandemics. And then therefore the question here is, what are the features that we should be looking for in selecting and developing a set of the most useful and suitable technology platforms to make widely available for outbreak response R&D in ways that also they deliver products that they can be equitably accessed everywhere. In addition to that, this is a series of is one of a series of webinars that will also inform other processes and other initiatives. For instance, there is a meeting that will be held at the Rockefeller Foundation in Bellagio in conjunction with the independent panel. There is a forthcoming issue, a special issue of vaccines that we're working on, but it will also be useful to inform the strategy and visions of a number of projects and initiatives, including the PSI. We have the pleasure to be having here 3 panels with the outstanding cast of panelists and 1 final respondent, and then the whole thing would be opening up for group discussions among panelists and questions from the audience. There will be quick introductions, and so I will quickly now handing over to the moderators for today, who are Els Torelle and Andrew Fowler. And if you have questions, you can use the Q&A session. I believe the chat question is not active, but a Q&A question for you to post your questions. So thank you very much, and on to us.
Thank you so much, Piero, for the introduction. And so we have a very dense program. So in order to save time, we also are not going to do extensive introductions, but we will put the bios in the Q&A. There's a link that Asia will share, so you can find everything about the different speakers today. So the first panel, we will really look at the centrality of adaptation and what makes a certain technology platform adaptable. What does it take? And I'm very honored to have as the first panelist that I can introduce Professor Dame Sarah Gilbert, I think known to most of you as one of the scientists behind the COVID Oxford AstraZeneca vaccine, and where we will we want to hear from her, because adenoviral vectors are not new. They actually have, and Sarah and her team have been working on many different ways and different indications to use these vaccines. And so the question we really want to hear about is, like, what makes this vaccine platform adaptable, and in particular thinking about rapidly adaptable and suitable for use also in low and middle income countries, and thinking already ahead about potential technology. Sarah, over to you, please.
Thank you. So I hope you can all see my slides now. What I'm going to be talking about for just a few minutes is the vaccine platform adenoviral vectored vaccines. And this is a technology that has proved its worth in the pandemic and shown to be a technology suitable for large-scale, low-cost.
You're muted suddenly. Is Eric, you're muted? We can't hear Sarah. Apologies, there seemed to be a technical problem around with the sound. Yes, I think, yes, you're back, Sarah. And can you put your slides on full screen, please?
So I hope you can hear me now. We're just going to get the slides onto full screen view for you. But I'm going to be talking about a technology that was used extensively in the COVID pandemic, adenoviral vectored vaccines. And this is a technology that has now proven its worth as being suitable for large-scale, low-cost manufacturing, also suitable for distributed manufacturing networks, and to produce vaccines that do not require frozen storage. They can be stored in refrigerated temperatures, 2 to 8 degrees, which is the usual storage temperature for most vaccines that we have in the world today. So this helps a lot with distribution of the vaccines when we need to do this quickly, because it means that we can feed into existing vaccine storage and distribution networks without having to make new arrangements for the vaccine to be stored, transported, and distributed. So I would really like to move to my second slide at this point, if that's going to be possible, but I think we might have a slight technical difficulty this end. So I'll just carry on talking about adenoviral vector vaccines. Move to the next one. Thank you. So the technology that we have in Oxford is ChAdOx1, which is chimpanzee adenovirus and OX1 for Oxford 1. But there are other replication deficient adenoviral vaccines that have also been used to produce many different vaccines. And the main features of these are very, very similar. So we are using a simian adenoviral vector, so an adenovirus that has been isolated from a chimpanzee originally rather than a human. And the reason for this is that adenoviruses are common human pathogens. They cause, can we go back up? They cause generally fairly mild respiratory disease, and many people will have been infected with different serotypes of the adenoviruses through life, and that means they will already have some immunity to those human adenoviruses, which has a limited extent, but it does to a certain extent reduce the immunogenicity of a vaccine which is made using the same human serotype. And this has been demonstrated with Ad5, for example. So to avoid that slight reduction in immunogenicity in people who've been infected with the adenovirus serotype, we use a serotype that they won't have been infected with because it's been isolated from a monkey. But generally, these the genetics of the human and the simian adenoviruses are very, very similar. So obviously adenoviruses are pathogens. If we want to make a vaccine vector, we have to make them safe. And we do that by taking out the E1 gene, which allows the adenovirus to replicate. It's provided in the cell line that's used for manufacturing so that the vaccines will replicate as they're manufactured. But once somebody's been immunized, that adenovirus cannot replicate. It cannot spread through the body. It cannot cause disease. It's also common to take out the E3 gene. That also gives us a little bit more space for adding in the antigens that we want to express when we make the vaccine. So in order to produce a vaccine from a replication deficient adenovirus, what we need to do is encode that antigen sequence within the adenovirus using a strong promoter such that after infection, after vaccination, somebody's cells become infected with the adenovirus, and that antigen is then produced inside their cells. This has a number of advantages. Firstly, that we don't need to worry about the stability of that protein antigen because it's provided in the form of DNA, as in an RNA vaccine or a DNA vaccine, and the protein antigen is produced only once somebody becomes vaccinated. And it's also produced inside a human cell, and it has a very similar structure, therefore the same conformation. It's not necessary to use pre-fusion stabilization, for example. We don't need to modify the structure of the coding sequence that we use when we're producing that antigen. And it's also not a structural part of the adenovirus that we will be immunizing with. So pre-existing immunity to the vaccine antigen can have no effect on the immunogenicity of the vaccine because the antigen is only produced after vaccination. It's good at inducing strong B and T cell responses, even after a single vaccination, but we've now demonstrated very effectively that we can boost those to higher responses with a second vaccination. So Chanox-1 was developed in Oxford, and before 2020, which was when we started the phase 1 studies of the COVID trials, we'd already vaccinated 330 subjects in early clinical trials, but more broadly, the CHAD platform had been used in 6,000 subjects of all ages. So there was a lot of information known about the platform. That means it's very quick to adapt. We just need to change the coding sequence of the antigen, but we already knew how to manufacture the vaccine, how to store it, and what dose we wanted to use it at, and what was the likely immune response that we would get post-vaccination. In particular, what was relevant was that we had conducted a clinical study with a MERS vaccine, again a coronavirus, and this was directly applicable to the development of a new coronavirus vaccine. So next slide, please. We started the phase 1 trials in 2020. We expanded our clinical trial network. We partnered with AstraZeneca for large-scale manufacturing of the vaccine, and they then set up a distributed manufacturing network. So I'm going to skip to the end of the process now. The network of manufacturers that was set up around the world meant that 3 billion doses of the vaccine were able to be delivered by the beginning of 2022. The aim was to have local supply for local use, to have the same seed stock and the same manufacturing process being produced around the world. And this is a really important thing to think about when we want to produce vaccines in a short space of time to immunize very large numbers of people. A distributed manufacturing network allows this to happen and was very important in the pandemic. So I'll stop there and come back to any questions that you may have for me later on. Thank you.
Thank you so much, Sarah, and apologies for some of the technical glitches in the beginning. Maybe if I may have a quick follow-up question now, and then we'll open it up a bit later, because one thing your last slide clearly shows is indeed that there has been this massive effort of technology transfer such that there could be production in many different places. Can you tell something about I know that you've done a lot of efforts in that training and technology transfer. What do you think is kind of like the main challenge, or what you would see need to see different to make that even easier?
I think what needs to happen now is for more manufacturing facilities to be available, and particularly in parts of the world where manufacturing didn't happen in 2020 and 2021. This had to be set up very quickly. And that meant that the manufacturers who took on the technology transfer already had to be ready to, with the facilities already built, with the staff already in place, ready to receive tech transfer. And that's why those particular manufacturing sites were selected. And unfortunately, it meant that no manufacturing was done on the continent of Africa. So now what we need to be doing is looking to expand the manufacturing network. And there's a WHO initiative going on to set up new manufacturing hubs, which will take on RNA vaccine manufacturing, but I hope that that will be expanded to other technologies in the future, to have a more even distribution of manufacturers around the world, so that next time we need to do this, we can produce more doses more quickly, but with a more equal supply across the globe.
Yeah, thanks for that. And we'll hear more about the mRNA vaccine manufacturing efforts that are ongoing. But so let's maybe move to the next speaker for now, Dr. Fabrizio Chiodo. You're working with an Italian research institute, but we've invited you to speak about the Cuban efforts to produce a COVID vaccine or multiple COVID vaccines, because indeed they too have used different but existing technology platforms that they have adapted to COVID. So can you please tell us a little bit more about that and about what made that rapid adaptation indeed possible? Thank you.
Yeah, can you hear me?
Yes, thank you.
Yes. So, well, thanks, Els, for the invitation. I will be very quick because I'm mostly curious about the discussion later on. So I've been working with Finland Institute in Cuba since several years already. And here I'm going to show you how a system that is based on a public biotech can be an option for the future, especially for low and low middle income countries. As you can see here, Cuba nowadays is the country that has administered more vaccines against SARS-CoV-2 per 100 people. If you look at where United States are and where high income countries are, and unfortunately where low income countries are in this kind of ranking, we have to admit that what Cuba did was, in my opinion, extraordinary. But for most of the colleagues, they call this the Cuban miracle. But actually, that's not a miracle, because you have to think that it's a country that is under a very bad blockade by the United States, and not only that, in the last years, they have explored the concept of public health system, very good public health system, in combination with a very solid public biotech to control infectious disease, to lower infection rates, and also, of course, to decrease the costs of hospitalization that will make the health system suffer. So in the last 20 years, several public Cuban companies, they have designed, developed, and produced almost 80% of the vaccines that they administer to their children. And among them, for example, several platforms and technologies against Streptococcus pneumoniae and Neisseria meningitis. So when the SARS-CoV-2 arrived, in addition to move all the tools that the public health system had, they also moved all the tools that the public biotech already had. So different public institutions in Cuba, CEPI, Finland Institut, Institut SIM, they collaborate all together to develop 5 vaccine candidates that now we have 3 authorized vaccines, not only in Cuba, but also in different countries. They are all protein-based vaccines. They are all based on the receptor binding domain. Of course, for some colleagues, they do think that, oh, this is a traditional way. We have to be also honest in some manner, because yes, but what do we mean with traditional? Because, for example, the conjugate vaccine Soberana 2 that is optimized to evoke B and T cell responses in children nowadays is the only SARS-CoV-2 conjugate vaccine in the world. So playing with these tools, Cuba have vaccinated also more than 98% of the children since 2 years old. Of course, all the data have been published. We have at least 15 publications in peer-reviewed international, very well-known journal. All the data have been always published in the medRxiv or bioRxiv database. Of course, the tools, whatever they are different between them, but I will enter in the detail later on. They are highly scalable in terms of production. They are cheap. I have nothing against mRNA vaccine. I'm a scientist. I do love these technologies, but we have to admit that, for example, for boosting our immune system and the B cell memory, for example, maybe a receptor binding domain boost would be for sure more stable at room temperature, like all the Cuban vaccines, and the costs are at least 10-20 times cheaper. But in addition, my key message to close this is also what the Cuban public system is as a hope for poor countries, and also as a hope to trust the public system. Because that's my last slide. Of course, at the beginning, Cuba had to suffer because of the blockage, because we did have all the ingredients to make the vaccines. We had 5 vaccine candidates, but due to the blockade, we didn't have the syringe to vaccinate the population. But then Cuba jumped like none of other countries did. And that was what, based on the receptor binding domain that we use, that allow the surface, a particular part of the receptor binding domain that nowadays also all the new Omicrons are still recognizing. But in addition, the trust of the people that were vaccinated, and so boosting all the population with this rate I do think that Cuba is one of the countries that was able to control the Omicron wave in a very elegant manner, respecting people, and of course, by using technologies already explored in the countries with a public biotech system that linked different institutions all together. So for now, I think I stay in my 5 minutes and looking forward for comments and extra slides I have. I'm here.
Thank you so much, Fabrizio. I think that it's a very nice illustration indeed of how these existing technology platforms can be quite rapidly adapted. And I think one other element which we learned from the Cuban experience is also that different constructs have been used in combination with different boosting than the primary doses, etc., which has been very difficult to implement in a very strategic way in other countries. So I think that there are some things to learn from. But for the sake of time, I would like to move to the third speaker of this panel, to Stig Tollefsen from CEPI. CEPI, the product development partnership that was set up to specifically develop vaccines for emerging infections, for outbreak diseases, of course. And you have, as CEPI, looked very closely into a multitude of technology platforms and what they can offer in terms of the possibility to rapidly adapt in case of an outbreak. And so if you want to share a little bit of your experience and specifically what exactly makes a certain platform rapidly adaptable and with a view of production technology transfer, what are the characteristics that you have been looking for? Thank you.
Yeah, thank you very much, and it's a pleasure to be here today. I thought I should talk a little bit about how CEPI operates. Can you see my slides? And are they advancing? Thank you. So we are a private-public partnership that supports development of viral vaccines where there is no commercial market incentive. And it was established in the aftermath of the Ebola outbreak in 2014. So what CEPI has been doing is collect money and then run open calls for proposals, recruit the best vaccine candidates, and then support the development of new vaccines. So I thought I'd show you just this slide of our portfolio with some of the vaccine candidates. They are mainly from the blueprint list of the WHO. So you will recognize the MERS, the Lassa, Nipah, Chick, etc. The last couple of years, we're working on the COVID-19 and the broad protected coronavirus vaccines. You'll also see the disease X. That's a particular mindset of CEPI to develop vaccines against the unknown disease that might come. So then we are looking for suitable technology platforms. And one example that illustrates how we work is I was in the office in London early 2020 in January. I ran into Richard Hatchett, our CEO, and he was rather concerned at the time. There has been some puzzling news from Wuhan. Some rare disease was going on. We didn't know anything about it. And then eventually, Richard was saying that this can be big. This can go really wrong. And unfortunately, we didn't believe him, but he was right. But the important thing here is that as soon as we had the sequence, we would start reaching out to our developers in the portfolio to ask if they will be able to start modifying their vaccine platforms to answer to the new virus that we had seen, the SARS-CoV-2. So the MERS developers were there. Sarah was one of them. You had some of the technology platform groups working on platforms that was easy and modifiable. So we could start early on by developing a vaccine. And I think that's one of the mindset of CEPI to prepare in advance. So although it felt like years, it took a total of 326 days before the first vaccine candidates got the emergency use license. So the question we ask ourselves in CEPI now is, can we do this faster the next time? So now we are aiming to, we have launched this 100-day mission. That means that you should have a vaccine available 100 days after the sequence have been published. And you could maybe do a fun comparison with the Formula 1 racing sports, where in the 50s it took 60 seconds to change one wheel on the car in the pit stop. But today it takes just 2 seconds to change all 4 wheels. And they have managed to do this by just changing or working on improving every step in incremental small steps, but in a total over years, you manage to improve the process and perfecting it. And that's also the CEPI ambition, to do as much as we can to improve all these steps. So we are searching to find innovations and improvements of vaccine technologies and vaccine platforms. We are also looking into the preclinical development. What can you do in advance to the established models, assays, to make sure that your vaccine works? Biomarkers for early detection of immune responses. What innovations can you do in the manufacturing? Either is the mRNA, the viral vectors, protein, whatnot. And then clinical trial innovations. So how can they be done earlier on? Can you use these biomarkers you identified to say early on that you really have a vaccine that induces immune responses? And then try to complete as many pieces of the puzzle as we can before a new virus emerges. So we have started to work on developing a library of prototype vaccines. So we have an overview of all the viral families, and some are more prone to cause outbreaks, epidemics and pandemics than others. So you could do some prediction. You could prioritize a few of them and select, and then start working on the immunogen selection and immunogen design, and test in advance in some of these preclinical models. Then you could look at what kind of rapid response technologies you have, the viral vectors, the mRNAs. So can you do something to improve the protein-based vaccines, and etc. We also aim, and Sarah touched upon this, to establish global networks of manufacturers. Could you establish a network of manufacturers in the affected regions, close to where the outbreak is? You need centralized lab to test the serum samples from both the preclinical studies and also the clinical studies, with using standardized assays and reagents. And it's good to have those in a network and also close to where the potential outbreak may occur. And then you would have clinical trial sites and trained personnel. What can you do in so-called peacetime to train these people and set up these facilities? That's also what CEPI is working on. And then in total, you also need to work with the regulatory authorities to have some of the regulatory processes in place in advance before the next outbreak. So I represent the CEPI Office of Technology, and one of our overall goals is to look for some of these innovations that might help us to develop vaccines faster the next time around. So we have a mantra looking for speed and scale. How fast can you develop your vaccine? How many doses can you produce in 100 days from an outbreak? Some technologies might not be suitable for a high speed, but can have a high scale, and vice versa. Then thinking about the effectiveness, how effective are your vaccine candidate? Do you prevent viral transmission? Is it a rapid onset of protection, etc.? So that's also another aspect of what we evaluate and think about. And then a final component is the accessibility. Could you do with a single primary dose? What's the cost of goods for a vaccine candidate, and also the thermostability profile for the vaccine candidate? And for CEPI, it's also important with equitable access. That means that anyone who needs a vaccine should receive a vaccine without independent of the ability to pay. And I'm happy to announce just today that it was recently published on the CEPI webpage that we have signed a contract with the 20Med. We're aiming to stabilize the mRNA vaccine technology through a lyophilization process. That's one of the programs we have recruited through our open calls for proposals.
Thank you, Stig.
Do I have one more slide, or 1 minute, or 30 seconds?
Yes, please.
Yeah, so this is the 100-day mission. It's very ambitious, and it's not for CEPI to do this alone. We need a global collaboration on this and work together. And with the current experience, we know that we need to do better next time, and the next pandemic might even come tomorrow. So that's really the message here: strengthen the global collaboration and work together on the next pandemic. Thank you.
Thank you so much, Stig. for the sake of time, because we are running a little bit behind, and I know that one of our next speaker, Amadou, cannot stay very long. I want to move to the next session, but just one question that I want to put out here that maybe we can come back to in the general discussion. That is, your presentation, Stig, was very much focused on going faster. And of course, we know that for epidemic and pandemic in particular, speed is of essence. But the feeling exists after COVID that it wasn't as much the lack of speed that was the problem, because we've never had multiple vaccines so fast. I mean, it was amazing already. We never thought that we would be able to do that, so we did. But where it went wrong was the equitable access, right? And so I really think that we should be thinking about what makes these adaptable, rapidly adaptable technology platforms also suitable for that equitable access. And so maybe if you want to think about that question and to see how CEPI thinks about it, because as you know very well, that has not been the easiest of all things, and we need to move that bit for the future. And with that, I want to hand over to my co-moderator, Andrew Farlow, to take us to the next panel. Thank you.
Thanks very much. Thanks, everybody, so far. The first person speaking, the next panel is Amadou, who is at the Institut Pasteur de Dakar, which has been one of the very few vaccine producers in Africa. Since COVID-19, multiple initiatives have been underway at IPD, including therapeutics, mRNA vaccine, diagnostic, and so forth. Amadou, can you speak to the vision of establishing the capacity at the IBD for multiple technology platforms to respond to regional health needs, and how you prioritize, choose your portfolio? And I'm thinking of the last discussion of the 100-day mission. You know, one of the dangers is we're already in 100 days with something, but it goes to high-income countries, or they queue up for it. How do you fit into a vision for equitable access, even if there's a 100-day mission?
Thank you very much.
Put his slides up. Great, thank you.
Could you hear me?
Yeah.
Okay, thank you very much. Just wanted quickly actually to show with the slides in terms of vision, that it's a lot about being early at the detection and the responder, because that's what we think it would prevent actually major catastrophe that we have for COVID. And really the second pillar is about all the time trying to innovate, bringing new R&Ds and educating people to build that. And the third pillar of vision is really the biomanufacturing, but mostly for the purpose of equity. What is critically important for us in that vision is really to be aligned with this dynamic that Africa is bringing by building 5 different hubs where we were planning to do more biomanufacturing in vaccine per se, where the purpose is really to move from 1% manufactured locally today, as Sarah was mentioning earlier, to 60% by 2040. So 20 years to do that in line with other experience that we see. In that purpose for us, the way to do it is really to build a platform on which we can receive other platform that would help us build the ecosystem that is needed. And this is typically actually the sort of platform that we built here in Senegal, where you have actually in these slides a first facility focused on one technology, which is egg-based, that we master over years by manufacturing yellow fever. A second platform around cell culture, which is really turning for using technology like what Univercells is doing. José, I think, will talk later about that. But also, besides the capacity to do manufacturing, to also have R&D, because what we realize is if you want to build a portfolio for the next vaccine, if you want to make sure we master some of those technology, we need not only to be very close to the field and make sure actually we have the right capacity to adapt very quickly. It's not just about manufacturing, but making sure that we can adapt quickly when we have new information to meet the 100 days, for instance, what CEPI was saying. And the real critical part of it is not only having the capacities to manufacture and R&D, but the critical point in Africa is to have supply chain and make sure you can deliver those technology. And that's where, for us, it's important not only to have this integrated platform to be recipient, but the capacity to work with others also to deliver those things. I just want to say something like partnership. I'm going to take this example where really what we're doing with a measles rubella vaccine, which is a disease important, facing a lot of shortages. There is this work where we use partnership, where we have technology from Univercells working with different groups, Batavia's with the process, Kansas University, to try to bring together within a very short period of time, actually a vaccine that would be done at very low cost. And the fact that we master this sort of technology would help us bring whole sort of line of technology, including the Charox that Sarah was mentioning, or the technology that José will probably talk in details. Through this sort of partnership, we can make that available as soon as possible. In those partnerships, obviously, we're working with the messenger RNA apps, being a good actually opportunity for them to transfer, like we're doing with Petro, and also making sure that CEPI vaccine can be actually manufactured in Africa for Africa through these manufacturing networks. I'm just emphasizing all these different partners because for me, besides having the technology and the capacity actually to do research to adapt it to the local context, it's even more important to have the business models and the capacity actually to deliver those things. In this line, we've started this with vaccine, and we are also doing diagnostic, and Joe going to talk about it later, and therapeutic maybe later. And the vision is really leveraging new technology through the platform and make sure whenever we have a new pathogen that would be built through the very strong surveillance system built upstream, we would be quickly have an opportunity to translate that into a product and later a product that can be manufactured for the sake of equity. I don't want to be long. I just wanted to lay that out and we'll be happy to take any question. Thank you.
Could you give us some thoughts? You use the phrase business model, which is kind of fascinating to hear. I mean, we know that we can't just transfer technology for current COVID-19 vaccines. The pandemic vaccines are not going to sustain any of these things, these new initiatives for the next 20 or 30 years. So they've got to deal with the interpandemic period, be ready for epidemics as well as pandemics, and build a portfolio of products. Can you give us a little bit more of the vision for that? I mean, at the moment, you paint a picture of lots of partnerships and resources and money coming in, but at some point, this has got to be self-sustaining, and it's got to keep going on the basis of changes in the ecosystem, the way in which I noted Gavi and funders work, a different model. Can you give us a bit more on that different model, that different business model even?
Yes, clearly. I mean, for us, building a platform like that would work with a business model where the objective is really to be financially sustainable. And in this regards, typically that capacity can be used as a CMO for the people that want to deliver vaccine for Africa. This is one of the sort of model that we do. Let's say Indian manufacturer, Korean manufacturer from other parts of the world may want to deliver Africa vaccine, but making it locally would be a huge advantage. Having the capacities to use us as a CMO is one way we can try to generate revenue, making it more sustainable. On the other hand, we can use our capacity, fill and finish, to be able actually to bring some vaccine for which capacity is the problem. Many of the players usually have capacity in pandemic context. You cannot actually deliver it because people don't have fill and finish capacities. And in these regards, we're trying a new technology that has been brought to us by CEPI preliminary work, where you can fill using pouch technology, a capacity to have 200 doses a pouch that you can use actually to vaccinate quickly and have large capacity up to 1 billion doses per year, which is very necessary if you talk about pandemic. And the last sort of ideas in terms of business model is obviously we have to build this financial sustainability to work with main players, like Gavi and global funds and main players to use the UN system to deliver large amount of vaccine. But if you want this business model to be sustainable and relevant to Africa, revenue generated by these large manufacturing should be also used to do less profitable vaccines and that are highly important for Africa, like Lassa and things. So for us, it's more like generating revenue using the highly sold vaccine to try to support the ones that are less supported because of the fact that they're touching population that are not in a position to afford those vaccines if they are too expensive.
Thanks, Amadou. Sorry we don't have more time to have you talk longer, but hopefully you'll be around for some of the question and answer. Thanks very much. Petro is up next. Petro, the big innovation in terms of vaccine technology platforms since COVID-19, some people say has been mRNA. My screen is playing up at this point. You're leading the first mRNA vaccine technology transfer hub, the WHO coordinated project that aims at building capacity in developing countries. Why did you choose mRNA technology for that particular endeavor, and what features makes this technology so versatile and suitable in your mind at this point in time? And are you thinking of moving on to other technologies?
Andrew, thank you, and good day to everybody. Can you see my slides, and is it on full screen? And can you hear me? Thank you, mRNA. Let's talk about mRNA. Maybe just to give a perspective, unprecedented achievement for the world in terms of the speed of vaccine development and vaccinating delivering more than 12 billion doses of vaccines in a period of 36 months. And it's clear if we analyze that it was significantly influenced and dominated by the viral vector, adenoviral vector platforms and mRNA platforms. And for mRNA doses, a quite significant contribution to COVID-19 vaccination for a new platform for vaccine market authorization, not a new platform for development of a product development. I want to encourage people who have not read the article on the tangled history of mRNA vaccines, the beauty of the science, the scientists, the players, the innovation, starting in 1961, the work, and I'm not going to go into those details, but really encourage you to read this paper, which is published in Nature in 2021. But let's talk about the mRNA Hub program. This program, launched by WHO and partners in early 2021, after the realization that there's insufficient vaccine supplies to low and middle income countries. So what is the vision of this program? Is to create capacity and capabilities in low and middle income countries. The significant decision that was taken strategically is to use an mRNA manufacturing platform as the platform for capacity building in those regions. And I'll go into the reasons for that. Those were decisions taken by experts in the WHO and in close consultation with many, many parties. What is also important in the objectives of this program, and I think it circles back to what Sarah has been saying, what Amadou has been saying, what CEPI has been saying, is also to stimulate regional research and development. And we are particularly aware of the importance of continued R&D in the mRNA space to address some of the target product profile issues, but also to advance the technology to be truly sustainable. And I'll mention a bit about that as we move on. Certainly, what is this program about? The principle, and I've highlighted here, is equitable access to technologies which are suitable to respond to pandemics. And mRNA technology has the properties and abilities to meet that goal. What is very important, and Amadou touched on it, is the issue of sustainability. How do we ensure that in the non-pandemic times, what we've built here is sustainable? And how can mRNA be used to do exactly that? So maybe a little bit about the unique features of mRNA technology: high precise, non-infectious, the transitionality, non-mutational, but very important, the rapid response capabilities, which is not unique. I think Sarah earlier clearly pointed out that there are other vaccine manufacturing platforms that are also suitable for rapid response. mRNA brings other unique properties and qualities to a rapid response combined with routine manufacturing capabilities. And this slide from Barney Graham that I think summarizes it very well. COVID-19 really demonstrated that mRNA vaccines can be safe and effective, and it is a scalable modality for vaccine delivery. The prototype pathogen approaches to pandemic preparedness makes it very suitable as a screening and a developer of multiple vaccines. Stability and supply chain issues is a challenge. It is improving. The CEPI announcement recently allowed them a stability. This mRNA hub already participating and working with Univercells and Etherna to also look at a lyophilized COVID-19 vaccine to demonstrate capability. Small footprints, small batch manufacturing, a different unit of economics, and certainly very suitable for low and middle income countries for transfer and also for R&D purposes. And you just say in the next presentation is really going to demonstrate what they have achieved and also working with us and with Amadou and putting technology, process technology on the ground that leans itself to a totally different economies of scale and will bring mRNA vaccines cost of goods down to affordable. We have to be really realistic. It's not the magic. This is not the technology which is a solution for all and everything, but it's a technology that brings enormous opportunities for innovation, but also manufacturing in low and middle income countries. The point on the network, this is a vast network that of the mRNA hub program. These are the countries and the companies that will receive mRNA technology from the hub here and consortium here in South Africa. What is very significant, if we look at these 15 companies, is the levels of maturity and the capabilities that already exist. Some of these companies will diversify their platforms, all low and middle income countries, diversify platforms using mRNA, getting access to mRNA technology for new vaccines and additional vaccine manufacturing. And others, like in the case of Nigeria, Kenya, will be greenfields developments, which will enable them to also have platforms for vaccine manufacturing. I'm going to stop here, but just also mention that we are aware of the challenges, the issues on stability, the potency issues, the cost of goods issues, and all of these are now intensely across the globe being addressed through R&D efforts to ensure that mRNA technology as a vaccine manufacturing platform is sustainable and is well embedded and contribute not only to pandemic preparedness, but to vaccine supply globally. Thank you very much.
I use the phrase "some say earlier" because, as you put up on one of your slides, you know, this isn't new. This technology has been around a long, long time, but this is only the third sort of product that was actually reached the market from it. Okay, my question would be, you know, this technology has had its success. It's opening up. Lots of new products are coming along. But so far, it's you know, the profits to the stakeholders in the private sector have been absolutely huge. Now, going on into the future, you're talking about epidemic vaccines that may not have a great market. You're talking about products for low-income settings produced by technology in low-income settings. How are these two systems going to compete against each other, or are they, or are they not? I mean, what's your long-term sustainability strategy? We know that the technology, now that the door's opened and lots of new uses are there, high-income countries will be looking to use the technology for cancers and other therapies. What do we need in terms of IP, institutional details, regulation? How do we protect the knowledge and protect the ability to share knowledge and data so that there's equitable access to the technology and then to the products?
Andrew, how much time do you have?
I didn't say do it in one sentence, at the very least. But we know that there are interests and there are challenges, and this is fantastic. I mean, we talked about it before. It's fantastic what we see going on here, and one or two of the other speakers will talk about it. But you do kind of worry at one point, when does it become sustainable, and what will be the pressures on the system, and how do we prepare and counteract some of those pressures now, as it were? Yeah.
So, Andrew, many aspects and lots of discussion. Sustainability will come through managing cost of goods. Sustainability will come through a pipeline of products. We have 2 registered vaccines utilizing mRNA technology. This is a phenomenal breakthrough in the history of 30 years of research. But we have a pipeline. If you today go and look at the pipeline of products been developed on mRNA platforms, and by the way, there are different mRNA platforms. It's not only, we need to recognize that. It's more than 700 in clinical development. And those are certainly, and I have slides on that if anybody's interested to see who's doing what and where is it. And this is the January 2023 data. But certainly there are legacy vaccines, there are neglected disease vaccines, and there are vaccines that exist that can be repurposed, and there is still not capacity in low and middle income countries to produce many of these vaccines. And if we can combine with models around economies of scale, carefully, strategically participate with the CEPIs and other parties globally, that we do not exceed demand, that we develop business models which are sustainable, that we have multiple products that can be produced on these platforms, and that with groups like José, who's the next speaker, that we actually get to below $1 a dose for the drug substance manufacturing. And that is certainly possible. mRNA is gonna address the challenges that is currently potentially limiting its value to public health. On the IP landscape, it will get more complex. It is a network of huge complexities and a whole day discussion on its own, but not to be necessarily the blockage for access.
Great. Thanks very much, Petro. I'm afraid we have to move on, but we'll come back to you in some questions. José?
Sure. Let me share my screen.
You've been asked to respond to the following. Univercells has distinguished itself as a company developing manufacturing solutions. So you're going to talk about processes and manufacturing for vaccine and biologicals that facilitate and accelerate local production of affordable products. And you're applying this to RNA vaccine technology. And we've heard already today of a number of partnerships with a number of the other speakers where you're partnering to share this technology. Can you explain what it entails and why this technology is suited for this purpose?
Absolutely, yes. Thank you very much for giving me this opportunity to explain what we do. And indeed, thanks to all the previous talks, it would make my task easier today, because you guys already mentioned many things that actually drive us or feed us in the way we develop our manufacturing technology. So Sarah mentioned, talking about adeno, she said for me, the first magic word is a platform. And indeed, the adenovector is a platform also from the process perspective. So if you develop a great optimal process to produce adenovectors, there is a high chance that the same process essentially will be applied for any new vaccine candidate, the same for RNA, of course. And so this is actually what drives us and the way we develop our manufacturing technology. So Amadou mentioned us in your Institut Pasteur circle as equipment or technology provider. But what I wanted to share today is actually the way we develop our technology. So in essence, our goal is never to develop a new equipment. Our goal is always to deliver a new process. And the way this process is designed is always driven by our end goals. And the end goals were already explained here. It's all about being fast, being efficient. So we want to, as much as possible, to decrease the level of capital expenditure that will be needed to establish yourself as a new producer. And you would like this to be a platform. So you want to remove as much as possible, any process parameter that will be product specific. And so this is actually, and today I will just share a couple of slides on the RNA platform. So the first piece of the puzzle we are working on, and we have this currently collaboration with Petro, her team, so with Afrigen, is about the RNA production itself. So we always start with what we think will be an optimal process. First, the process is redesigned to avoid scaling up. So we always work in intensified conditions for the volumes to be as low as possible, as small as possible, in such a way that when we develop a new product, actually the phase 1 clinical material, the phase 3 clinical material, and the commercial material will be produced at the same volume. So there is no scale up. So no risk related to scale up, no time needed to scale up, and no associated costs related to scale up. So then this process will be delivered into all the mixes of reagents. That's the beauty of RNA from the process perspective, is that at the end, nothing is simple, but this is just enzymatic chemistry. You don't need to deal with complex cell culture. We do that as well, on other technologies. But here you have a great advantage, because this is complex chemistry, but still just chemistry. And then this is implemented into disposable reactors and consumables operated by an equipment. And so I don't talk about the rest today, but we are doing the same to produce synthetic DNA, which is needed, to feed the RNA, so the in vitro transcription. And we are currently doing the same for the scaling up of a process, to produce lipid nanoparticles. And so basically on this slide, I just repeat what I said. I just wanted to focus on the little square, on the left side of the slides, just for you to make concrete of what I was saying. So in this square, you see that on more than 80 different IVT reactions, both for the synthesis of RNA and its purification, using 18 very different RNA constructs from 1,000 to 11,000 nucleotides. So the last being a self-amplifying RNA construct. So using COVID-19 strains, using rabies strains, using other proteins, we have been able to optimize the process with this goal in mind, is to make it RNA construct agnostic or independent. And so you can see that for all in the experimental domain, we have defined and optimized. Today, at least for vaccines, I'm not talking here about, ultra highly pure RNA for cancer, for example, but we are, below all the existing specifications in terms of residual, double-stranded RNA, in terms of residual uncapped RNA, residual proteins, residual DNA, with a yield which is always above 5 grams per liter, and a capping efficiency that is always well above 90%. And so this is actually, the essence of what we develop, of what we want to provide for this technology to be actually fast, in the sense that the process is pre-designed as intensified and chain. It's the same, exactly the same conditions for all the constructs I was referring to. No scale up is needed. And so this is what then we deliver into. You see here a picture of actually the system that will be shipped to South Africa in March. Actually, it is now being qualified into a portable container that you see here. So the machine you see here basically implements the process I was referring to. So the main benefit is the development, the process development is totally de-risked. So you gain in speed. So in terms of pandemic response at very low cost of goods, we are still going that effort with Afrigen now to compute precisely all the drivers of cost. And this is delivered into a very small footprint, addressing the capital expenditure issue. You see the machine you see here will be able at full capacity to produce 10 kilograms of RNA per year. So depending on the dose, this might be equivalent to 100 million doses in a container of 25 square meters. And of course, the same system, the same process, the same machine will be actually also suitable to produce the first phase 1 clinical material. I will stop here and take any question.
In our report that we put together for this vaccines journal, one of the co-authors used the word ecosystem, the need for an ecosystem into which this is embedded. That is scientific, clinical trials, regulatory and commercial interests. Now, when you send a container to a setting, it arrives there and it needs lots of other things apart from what you're doing. So I'm thinking regulatory, monitoring, quality control, supply chains. Can you give us some insights as to some of the things that other players, not just companies like you, but other players should be trying to put in place to support your activity?
Yeah, sure. And so actually you gave the answer. So in this, I think this is a great piece of the puzzle, but it's just one piece of the puzzle indeed. And so, training of workforce is fundamental. You need, the recipient to be trained, not only to this specific technology, but to be trained to vaccine development. And so that's, for example, one of the reasons why we like this initiative of WHO is putting that network together, putting together all those competencies, working with, your organization, Stig, like CEPI. So I think that training is a must. Without that, this will definitely be a failure. And training in process, in vaccine development, in immunology, in regulatory affairs, in clinical development. So there is a massive effort to be done. What we provide here is just a piece of it.
Great, thanks very much. We're conscious of the time, and I've been told to push over to Els, who's going to do the next three speakers. Thanks very much, José.
Thank you so much, and yeah, it's already a very rich discussion that we're having, and we want to make it even a little bit more complex to move also, because so far we've mostly talked about vaccines, but of course, in terms of epidemic response, we need multiple health technologies, and even though there is a lot of power in vaccines, without treatments, diagnostics, it's just not good enough. And we want to extend that concept about the platforms, because I think it came out of many of your conversations, like the value of a platform that is versatile, that can be adapted quickly, etc. And actually on diagnostics and certainly some treatments, we have some platforms as well that we want to, at least for this afternoon, highlight as an example. And we'll start with Gary Kobinger, who will talk to us. He has done a lot of work on vaccines, including the emerging diseases vaccines, when he was working in public health Canada. But he has now also worked a lot on monoclonal antibody technology, which has grown into one of those new technology platform with a lot of promise. And so, Gary, over to you to talk a little bit about what that technology can do. And especially, it used to be very expensive and kind of reserved for niche indications, but a lot has changed again in terms of technologies that can make them more suitable for this type of applications. So please, over to you.
Thank you. Hopefully you can see the slide and you can hear me.
We can't see the slide. There is, we only see a little piece of your screen. Can you try again, because it worked earlier?
Try this. This is good?
Yeah. If you can put it on full screen. Yeah. Thank you.
Oh, you have it on full screen now? Maybe not. Huh?
Yes.
Sorry about that.
Try again, because sometimes it's a bit slow. Oh.
No, the issue is I'm having two screen now, and that's why it's a bit different. The setting. Let's see again. Well, we'll do it that way. This is working?
Yes, perfect.
Okay, thank you. So I'm going to be fast. I'm not sure which one I need to move forward, but I'll do both. So I wanted to have a more like economic approach to what has been presented and going forward and focusing on MABs. But just quickly, this is the list of pathogen of interest, neglected pathogen of concern that were established first in 2012 and carried along the way, and it's going to be confirmed soon again, probably about the same list, and about budget that have been attributed to those pathogen. And just highlight, long list, that for now, what we have out of this is licensed Ebola vaccines, licensed treatment against Ebola with monoclonal antibody, none of which are available in Africa where the virus is endemic. Everything else, over $1.55 billion has generated about not much. I'm not counting the money for SARS-CoV-2, which was a huge success. That alone is well above $1 billion. I think that I need to absolutely highlight something, because I'm hearing, I think what I'd like to highlight is the importance of having flexibility and different options, different tools. In this case, in this context, what has been said when we were talking about adenovirus is absolutely important to keep, and recombinant protein, to keep a lot of tools, because keep in mind that a lot of those viruses, let's talk about filoviruses. If you have breakthrough infection, the benefit of vaccination is very limited. It will lead to death in animal 90% of the time. So now ask around you who has received more than 2 doses of coronavirus vaccine, SARS-CoV-2, and got infected. So what we are now thinking is going to work for everything in terms of protection may not work for everything in terms of protection in humans. So putting all your eggs on the same basket is a high risk, has always been. Is this moving forward? Do you see the next slide? Yes. Okay, thank you. So for MABs, what are the economy of MABs? Currently, production cost is at $95 to $200 US a gram. Actually, this is what leads to a treatment that is costing $10,000 or less, or about $10,000 or more. This is for infectious disease. By the way, for oncology, same amount, actually a bit less of monoclonal antibody usually are used, and you're about $100,000 extra. So what makes the difference between the two is definitely not cost of production because they're equivalent. It is profit margin. At $10,000 a treatment, this is why there's no treatment being stockpiled in Africa, by the way. And here again, the profit margin is massive. $95 to $200 US a gram is about the cost of production at small scale for doing clinical trials, but definitely not that large scale. You see the numbers here, and for insulin, for example, we're talking about $25 to $70 a gram for a recombinant protein, antibody being a recombinant protein. So there's a lot of room for improvement on the monoclonal antibody side. It's an emerging drug molecule that has been growing extremely fast in the past 5 years alone, but definitely even more so probably in the past 10 years. It has a lot of promises. When you think of it, it showed very encouraging result against SARS-CoV-2, despite the fact that it's a respiratory virus. So when you do an IV injection of a monoclonal antibody, it's not going first in the lung, I promise you that. So you can expect also a very high potency from monoclonal antibody from systemic injection of viruses that are disseminating systemically primarily, as opposed to respiratory viruses. Another thing I need to add is that, right now there's a lot of effort, for example, for developing a vaccine against Lassa virus, the uptake of which would be, has to be seen, very questionable in my view for being on the ground for years and years. You may have the best Lassa virus vaccine and may not be taken at all, even at $10 or $20 a dose. But one thing that could be useful tomorrow is a treatment that would save life immediately. And a treatment based on monoclonal antibody that should cost no more than $200 a dose, or a treatment is in my view, a priority. So I just wanted to put vaccine dose of what it should cost. The cost of good is $1 to $3. It should cost $2 to $3 and for the end user. For COVID, it's sold $20 to $30 a dose. And now the market estimate, when you see Moderna announced that they wanted to sell their dose, each dose $120 to $130 US. I'm sorry. So the goal for MAB is to reach $20 a gram. I think it's feasible, and the vaccine $2 to $3 a dose. How do we do this? There's example on the left of their data. On the top left panel, the CE8, the circle on the top right of this graph is a new monoclonal antibody that recognized more than 95% of CCHF strain that are circulating within different clades. Very promising. At the bottom, a single monoclonal monoclonal number 100 here that could inhibit virus replication of both HMPV and RSV. One single monoclonal. One against RSV is comparable to pembrolizumab, and against HMPV, actually the best that we have seen on the market. That monoclonal costs $100,000 to isolate. And now clinical development should not be more than $5 million, in my view. The top costs $300,000 to isolate because we had the big challenge of several clades to face. Next slide. So how do we get to that? Well, not-for-profit. Not-for-profit creates good job and can still make money. By the way, this is why I wanted to show this slide. It's just an example of a not-for-profit that I know because I created it in 2018. And not only it has great project ongoing, but it made money, and with the profit that had to be re-injected, it funded the clinical trials in part in Turkey that is upcoming against CCHF using favipiravir. This is Tom Fletcher's trial. So it can make money, can create good job, but most importantly, it keeps the cost down. The cost is less than 10% of every other pipelines I've seen. And we have experience all the way to phase 2 clinical trials. So I think these platforms are important. By the way, and this is not the only. A great success actually is Oxford, in my view. It's a recent great success of a not-for-profit. The mRNA, I think, effort in Africa from WHO is massively significant as well. And again, think about, this is just this session. Think about the success from Cuba with their vaccine, also using the same concept. So I wanted to show you what you can do for less than $10 million. Well, the Ebola vaccine was brought all the way to phase 2 clinical trials for $2.6 million. That included the clinical lot. The monoclonal antibody ZMapp, that was the fingerprint of what followed after that, cost $1.8 million, including a clinical grade lot for a phase 1 clinical trial. Now, more recently, again, I said it, RSV, HMPV, at the end was only $100,000 for the monoclonal antibody. I'm passing the rest, but with very small budget, you can move things forward in the context of not-for-profit. And are these?
You to finish because we're running out of time. Thank you.
I'm sorry, this is the last slide. I just wanted to show you how significant this could be. On the left, these are human data from the clinical trials that we just completed in Geneva. And what you can see is after a boost to the VSV vaccine using a DNA vaccine, the boost effect is long-lasting all the way to week 24, which is the first time that we've seen that, which has not been described before, not including in the New England Journal of Medicine paper that just came out. I'm going to pass the rest. Thank you so much for your time.
Thank you so much, Gary. And it's clear that each of these topics requires or could deserve a full presentation. We unfortunately don't have so much time. I just want to pick up on something you talked a lot about cost of production, etc. And I want to actually couple back to José, if I may, because you have worked also a lot, including on monoclonal antibodies and protein production. And I remember that you've told me once, and if you can make that point again, a lot of the current thinking around cost and cost of production relates to building economies of scale. Large-scale production will be cheaper. But I think, and you've shown it in your previous presentation, but also on monoclonal antibodies, etc., that's not always true, that there are now technological innovations that you can do to actually make small or small to medium scale production extremely efficient and economical. Can you say a few words on that?
Yeah, absolutely. And Gary said it in a very clear manner, and I totally agree. What drives the price of antibodies is not the cost, it's the margins. So that's very clear. Of course, I can totally understand if you need to produce, 3,000 kilograms per year, you need to invest, $500 million. You need to depreciate that. It's obvious. But for most of the applications or the needs we are talking about today, actually you need a small unit of 200 liters. Gary's developing, many others are working on highly optimized processing methods in, high cell density, under perfusion, continuous manufacturing. So your capex, can be in the range of, 15 to 20 million, and you can produce, massive amount of antibody for regional use. And there, indeed, the real thing is what Amadou was saying initially. What is your business model? If the business model is global health, you can provide many things at a very, very affordable price. If your business model is making money, then of course it's different.
Yeah, thank you, José, for reconfirming that intuition that we have, but that it's very good to hear from people who are actually building companies around that different business model. And so that makes me move to the next speaker, Joe Fitchett, who will talk a little bit more about another technology platform, but for diagnostics. Joe is working with Amadou in Institut Pasteur Dakar, where they've created Diatropics, which I think is a quite unique model of indeed local production of diagnostics. And so please tell us a little bit more about that and what makes indeed that rapid adaptation which you've implemented for COVID now, again, what makes that possible, and maybe also something around this economies of scale, instead of scaling up to a large, to one large facility, the kind of multiplication that you have been exploring. That would be very interesting. Thank you.
Thank you very much. And I think it's a good segue from Gary's talk around what small or relatively small amounts of financing can do for epidemic preparedness, because the facility that we established here really, thanks to the support of FIND in Geneva and Fondation Mérieux, IRD and others, Institut Pasteur de Dakar, was really sparked with about $1.2 million for 40 staff, and allowing us to have a focus on areas that had been neglected in our setting. So we're very pleased to be able to share a little bit about that with you. Just to start, this is a simple history of the Institut Pasteur de Dakar. It's been in Dakar for about 100 years, and in Senegal since the end of the 19th century. And vaccine manufacturing actually started in 1937, 10 years after discovering the yellow fever virus. And it took, as you can see, many decades to be able to then move to production of another type of biomanufacturing or health product, despite it clearly being useful and relevant for the region. So I think this highlights the importance of the COVID moment to make sure that regardless of whether mRNA is the right technology, regardless of whether protein subunits could be better, what we need to do is make sure we create those systems and do it in short amounts of time. For us, diagnostics fits in within the wider innovation roadmap, and great to see many of the partners at the table here, Gary, Petro, José, and others. And this applies as much to diagnostics as it does to vaccines. In our case, IPD is Africa's only manufacturer at the moment of both vaccines and diagnostics. And clearly, if the systems can be improved and elevated, there's a role for the next technologies. And really, it's around connecting the dots, which allows for this to work. And I noticed a question from Peter van den Huysden on the chat asking about academic groups, and we completely agree. Institut Pasteur de Dakar in many ways is an academic group, and we feel that that is something that has allowed us to connect those dots, because we have laboratories, we have sequencing capability, we have brilliant young scientists, and really it's the systems that we need to improve, at least to create the capability. And then when it goes to scale, of course, that's a separate and important challenge. If we look at genomic surveillance in Africa, we do think this is something that is really going to transform the response of the future. If we can put systems in place that can convert that knowledge, convert that material that is generated in key institutions into products. And you can see that in the right, in terms of volume of sequencing, the Gambia, Senegal, Kenya, Nigeria really managed to do a lot of sequencing under some pressure and with limited resources. Where do these samples and sequences come from? Well, we have, in our case, a Sentinel surveillance system. It's a fever syndromic surveillance system specifically, and it's distributed across each of the 14 regions of Senegal. It's being scaled up to go to each of the 79 districts of Senegal. And there are patient engagements of about in excess of 200,000 per annum for a population of 17 million. So a really important early warning system here that allows us within 7 days of fever at the clinical site to have a laboratory reference confirmation in Dakar in the central labs. But we've now started decentralizing some of those labs. And on the left, you can see the kind of infections, just the viral outbreaks that we picked up in 2020 and across these cities. And despite COVID, this kept going. And so this is really something that's very important for the development of diagnostics, as well as other countermeasures. And so what is Outbreaks? Well, it's a manufacturing facility dedicated to epidemic and neglected diagnostics. It kicks in when tests are not available, when tests are available but not satisfactory in their performance, and when tests are available, satisfactory in their performance, but really not affordable. And it's by design that the portfolio of the Outbreaks rapid tests is very matched to the strategy of what we have around the vaccines. So things like yellow fever, measles, rubella, filoviruses, these are regionally relevant. And as Uganda demonstrated the outbreak recently, you do not need, although it would be preferable, to have vaccines to end the epidemic. We do need diagnostics as well. So having access to both of these is key. And treatment, unfortunately, is very neglected when it could have a major impact on those that are sickest, something where we have a lot more work to do. Here's the team led by Chef Terblanche. This is the team that first started it with 4 or 5 staff. Now there's about 40 staff, and it's scaling up to a medium-sized facility to be able to start looking at targets like HIV rapid test production. And this, to talk about platforms, it really started around dengue virus, because the question that was posed when this was being set up was, what would a new facility in a region that has never had this kind of capacity focus on? Well, here you can see the outline of the unit. The unit chose to focus on things like dengue virus, hepatitis B, which are widespread burdens in the region, but also neglected threats, things like Ebola, Marburg. And then, I guess how we could call it is, major inequities in products around measles, rubella, and yellow fever, where those are 2, for instance, of the first rapid tests made for those pathogens, when clearly they cause a huge amount of harm globally. So here, just to end, maybe just to complement what Amadou was showing with the cartoons, here's an image in September. It's progressed quite some bit now, but gives you a sense as to how things can move fast when the commitment is there politically, economically, technologically, and with good partners, many of you around the table here today, and you know who you are. So thank you from us, and look forward to the rest of the discussion.
Thank you so much, Joe. Maybe a quick follow-up question, and I mentioned it already. Like in previous discussions we had, you were explaining also this concept of scale as horizontal scaling versus what is typically done is building one big facility, and that certainly the lateral flow diagnostic platform lends itself very well to that. Could you speak to that a little bit?
Happy to. So for us, certainly with some of the pathogens that don't have good, reliable tests, it's not as quick as, say, the mRNA technology, but you can pivot at very low cost to be able to start at least testing prototypes for good antigens and good antibodies. And there's so many reagents to choose from. Really, it's in that mastery of the platform that allows you to develop things quickly. Yellow fever is a great example. It took 6 months for the team in collaboration with the UK and Senegal to get the prototype done and to be demonstrated to be good. We had no guarantee it would be good, but we thought we could get there. But partners have been looking for many years to be able to do it. What was lacking was a facility. And that's really the value of a platform. It doesn't guarantee success, but if you're familiar with it, you have good access, like in our case, to a biobank, well characterized since the 60s, to clinical sites, to manufacturing partnerships, even if they're at small scale, as you saw, very small scale. That's the value of the platform, and that's the approach we've taken to vaccine as well. Our vaccine facility for yellow fever is very small. It's going to that giant facility we've seen here because of the advances in technology and because of partnerships with groups like Univercells, who José and team have been inventing new ways of producing en masse at low cost. Couple that with a foundation that's not for profit, we're starting to see movement. Sadly, not for the first pandemic of COVID-19, but we expect for the next threat.
Thank you for that. And with that, I want to move to the last of our long list of panelists, Achal Prabhala. And you have been working on challenges around access to health technologies for many years, and lately you've been looking in particular around towards regulatory challenges. And I mean, several of the previous panelists have mentioned already the importance of having the regulatory capability in the countries where these new technologies are being promoted, set up, etc. And some of the technologies are also new, which poses additional challenges, certainly in countries who do not have such a deeply experienced regulatory system. So yeah, I mean, could you just comment on what you've heard? And it's, again, a much broader conversation, but what are for you the highlights that need to be thought about after hearing all the different technological, process and progress and challenges that people have listed?
Thank you, Els. I have deliberately unmuted myself so that you can hear me speak instead of inadvertently unmuting myself when I don't intend to have you hear me speak. But the presentations were excellent, may I just say before I begin. And what I want to introduce is a provocation that connects to, I would assume, almost every one of your excellent thoughts and ideas that are based on your excellent work. And that provocation is around drug regulation that covers vaccines and biologics. And the simplest way for me to set this up would be to say, let's take a small molecule that we all know very well, the antiretroviral drug. So the antiretroviral drug, for about the last 20 years or so that we've known it in generic form, is very easy to replicate as a generic. The cost of process innovation or development is very low. The time to bring it to market, which is to say the time it takes to develop that and then have it approved with a local regulatory authority, is very quick. As a result of which, when it comes to small molecules, there's a clear and easy pathway by which, at a fairly low cost, a large number of competitors, generic companies, can produce said small molecule and bring it to market feasibly. When it comes to biologics, for instance, we just heard about mAbs, for instance, monoclonal antibodies. Now, to give you an illustration of why this matters is that all of the mAbs that you took in the countries that you live in Western Europe and in the United States are completely unavailable in India. So whether it's Evishield or whether it's the Regeneron mAb, none of those were available in India, despite us having an enormously large number of biologics manufacturers. And one of the reasons is that the regulatory pathway to a biosimilar, to a generic biologic exists, but it's tough for two reasons. So one, it costs a lot of money and it takes a lot of time. Why does it take a lot of time and why does it cost a lot of money? Partly because one of the requirements right now in the biosimilar pathway is a comparative clinical trial. Now that requires buying huge quantities of the originator biologic, putting that into something similar to a phase 3 trial, condensed into what they call a comparative clinical trial with your biosimilar product, then presenting the studies from that trial to the drug regulator in order for approval. That can cost several millions of dollars, sometimes near $10 million or more, and can take several years, which is why when you look at the number of biosimilars that exist today and that are in use in comparison with the number of generic drugs that are in use simultaneously with the originator version, the ratio is incredibly skewed. There are very few biosimilars in comparison with the number of biologics that exist in the market, right? So it's a tough pathway, but it exists. But now let's get to vaccines, and there's no generic pathway. We do not have generic vaccines. We have off-patent vaccines that have been around for years, which have no monopoly surrounding them on intellectual property, which have established what are called immune correlates of protection, which allow a vast number of manufacturers to make them. But when it comes to on-patent vaccines, new vaccines, right, of which there are not that many, by the way. So let's take the pneumococcal conjugate vaccine or any of the COVID vaccines. You either have the originator vaccine or a licensed vaccine, not a generic vaccine, right? And the reason is because there is no generic pathway to having a generic vaccine. And that's a problem. And I think it's a problem that we haven't understood well enough or sometimes haven't paid enough attention to, which for me, particularly living in a country with a large number of pharmaceutical manufacturers, is an enormous problem because it limits the amount of autonomous work that can happen in a country like India or any number of other countries on the African continent, in Latin America, other parts of Asia. And when it comes to mRNA vaccines, I think it's particularly egregious. I worked on a report with a colleague, Ala Al-Salhani, about a year ago, identifying over 100 facilities that could potentially produce mRNA vaccines using the proxy of currently producing what we'd call a sterile injectable, and not just producing a sterile injectable, but also having exported that sterile injectable product to either the European Union or the United States, or having it pass through the WHO pre-qualification process. So even with those fairly high quality standards, we found over 100 companies that would qualify on paper to be able to produce an mRNA vaccine across Asia, Africa, and Latin America. Now, the excitement with mRNA vaccines, of course, was because of the fact that they employ a biochemical process rather than use the traditional biology that we associate with vaccines, which meant that the production of them could be opened up to more than just the makers of traditional biologics, traditional biological pharmaceuticals. In fact, the difference we found was fivefold. So if we applied the same criteria to adenoviral vector vaccines, for instance, there would be 5 times less the number of facilities that could manufacture an adenoviral vector vaccine in any of these continents. And so there was something really exciting about the production possibilities of an mRNA vaccine. And yet there's a really vexing regulatory issue there, because the regulation for mRNA vaccines has not changed in any way whatsoever. So the regulation is set up to evaluate a biological product, whereas in fact, the mRNA vaccine is something in between a product of chemistry and biology, right? Because of the fact that it employs a biochemical production process without the use of the cultivation of cells or cultivated in eggs or in yeast as a traditional biological vaccine would employ. Which is all to say that certainly we need to look at regulation. But look, the encouraging trend here is that the process for biosimilar approval has just entered a new phase where the requirement for comparative clinical trials is moving to be dropped by using markers of equivalence, such as a pharmacodynamic studies, pharmacokinetic studies, that can establish a better and easier marker of equivalence without having to employ clinical trials over again, right? So there's some reform process that is underway in the approval of biosimilars, the regulatory approval of biosimilars. There's really none on vaccines. So either biological vaccines or especially for mRNA vaccines. And that's a shame, because I think that what it constitutes for me, I work against monopolies in pharmaceuticals. And what the regulatory system as it exists confers is a set of really almost intractable monopolies. We don't have laws that can overrule them, like, you have laws that can take a whole patent estate, for instance, and say, let's compulsorily license that. But we don't
can I ask you to round, please?
Absolutely. So we don't fully have a way to deal with the regulatory monopolies that exist with biologics and vaccines. And I think for any one of the wonderful solutions and options that have been presented here on the table today to exist, we need to find that way.
Thank you so much, Achal. And I think that the point that you're making is also really to say, well, I mean, regulatory pathways must be designed for public health, to protect public health, and if there is and to make public health means equitable access as well. And so if we now have a regulatory pathway or a lack thereof that stands in the way or becomes a barrier to equitable access, we need to kind of rethink the regulatory pathway to continue to protect, of course, public health as it should be, but also to promote equitable access. And I think that there's a lot of work that needs to be done. I'm going to hand it over back to Andrew. We are running a little bit late, so we have like 20 minutes or not even left for some conversation. I think there are a few questions in the Q&A as well, if you can try to combine that into a conversation with all our panelists. Thank you.
Okay. I've looked at the question and answers. First off, I just want to reflect on one thing. I mean, the topic today was key features of technology platforms. And then we were sort of thinking, how do you share those? How do you move them around? How do you make them? And time again, you guys drifted in the direction of saying, talking about innovation. It's about innovating on these platforms. Repeatedly, there was things starting with Sarah, you know, this mustn't be frozen. And then we moved to Fabrizio, you know, very heavily constrained system, and we had to innovate within those constraints. And then we had José developing in technologies to make vaccines that could be at a room temperature, at a fridge temperature that had historically been really, really cold. And then I think Gary hit on it. He said, you know, it depends what your objective function is. If you're innovating with global public health as your goal, you innovate in a different way. Whereas if you have a model that's innovating on the basis of big pharmaceutical profits and so on and so forth, it doesn't matter whether you're selling at 20 or 30. It doesn't matter what the cost of goods are, because you can sell at 20 or $30 a dose or whatever. And time again, you guys were pointing out that this is all about you guys innovating in your particular spaces. So I wondered whether you could have a debate amongst yourselves on, you know, how can we create a sort of really supportive, innovative system to support innovation. And this means also affecting the behavior of CEPI. I see you're there, Stig, but you have objective functions and you put money in particular directions. Gates is kind of fixed in its particular mindset. Governments are fixed in a mindset. How do we move towards where the objective function is global public health, and that starts to ripple through and affect all this innovation and enables you to do the innovation that you're talking about? So I'd like, if anybody wants to make an observation, perhaps Sarah could talk, and we're in Oxford, so my second sort of related question to that is, how do people like us who work in academia and in research centres, how do we support this kind of innovative capacity? Sarah.
Okay, thank you.
Brilliant.
So just to clarify, the adenovirus vectors can be frozen. It's a big advantage they don't have to be. It means
Yes.
it makes them much more
Yeah.
easy to distribute. What I think we can do to support this is working with the network of manufacturers that are being set up, and maybe even bringing more existing manufacturers into that network. So that I mean, what Acha was talking about was regulatory issues, and that's really important, because we have to be able to get these vaccines licensed and then used in different countries. And the way that AstraZeneca did that for the COVID vaccine was to have a network of manufacturers manufacturing a vaccine that was recognized by the regulators as being the same product. Because although it was manufactured at many different sites, it was done with the same seed materials, it was done with the same process, the same testing, and it was the same product. And that meant that we didn't have to relicense the vaccine that was made in every different facility. Slightly different arrangements with Serum Institute of India, who were a sub-licensee rather than a member of the original manufacturing network, but they were such a large manufacturer and they were supplying through the COVAX facility that could be managed as well. And so once we had MHRA approval, EMA approval, then WHO qualification, that opened the way to these vaccines being used in many different countries around the world. And I think we have to be very careful with setting up more manufacturing facilities and thinking about making new vaccines and new versions of old vaccines, that we don't end up with lots of duplication of effort, because every new vaccine that's created from scratch is going to need to be tested, it's going to need to be licensed, and that's not the efficient way to do it. So when I'm thinking about innovation as an academic, I'm thinking about innovations that we could then feed into a large network of manufacturers rather than just licensing to one manufacturer. So anything new that we do with the technology, I'm very interested in new routes of delivery, for example, something we haven't had time to talk about, but mucosal immunity is going to be a really big topic. If we can make improvements, if we can find new ways of doing things with a platform technology, we could then make that available to the whole of the manufacturing network, rather than just have one group using it and everybody else have to work it out for themselves.
Yeah. Can I throw the duplication comment to Petro? I know you've talked about it before. As you build networks, you've also got to manage it so that they're not sort of treading on each other's toes. Could you give us some thoughts on that?
Yes, and I think the word is probably captured in the phrase sustainability. You know what Sarah is saying here, if we look back in 2021, the AstraZeneca Oxford model was absolutely perfect. That's what the world wanted. Did that create capacity in low and middle income countries? Maybe not, but that model leans itself to doing exactly that. My view on the platforms is simple. The platforms, the different platforms are not the end. They are a means to an end. And I don't believe that any single one of these platforms will be suitable to meet all the demands in terms of vaccine and therapeutics. And again, mRNA leans itself to therapeutics and therefore leans itself to other innovations. If you have the ability to master the platform, you will also have the ability to also move beyond vaccines and into therapeutics. In terms of innovation, we need to innovate around the business models. We're doing exactly that. If we look at the structure here built in Cape Town, it is unconventional. It is small. It takes into account economies of scale, but also bankability. We need to innovate around how we make partnerships. We need to innovate in terms of where in the value chain do we actually enter. Do we enter at full finish or do we go back to drug substance? Do we create R&D capabilities to innovate? And Ashal, your comments around regulatory and vaccines is exactly what we are grappling with in terms of mRNA. Where does it sit? If you look at process, and these debates are happening. If you look at process and the fact that if you look at Quantum's process, it's cell-free. It's a completely synthetic process. There is no reason why this should be subjected to a regulatory process that will take 10 years and highly costly. It could be the hybrid. And I think there's some significant innovation also required in regulatory systems and thinking without compromising safety for people receiving these products.
Then Fabrizio.
So yes, I wanted to add.
Fabrizio first then.
Yes, okay. I just wanted to add a comment, and because I didn't have the time, and most a general one, you know. So I start from the academic, no, and then I start also to work with a public company from a poor country in general. So it's hard to comment on what do we mean when we mention innovation. So because something is innovation during a pandemic, and I totally agree, mRNA vaccinations, etc., etc. But then, of course, we need innovation when I think about cancer. And this is where mRNA will, I think, will make a breakthrough, hopefully for everyone in every society. But wait one second. What do we mean with innovation when we do have emergency like pneumonia, the killer #1 under 5 years old, and also against antimicrobial resistance? Because for these capsulated bacteria, all of them, mRNA vaccines do not work because this is biology. So I make vaccines with capsules. I'm a carbohydrate scientist. So we have to be fair with the society that the killer #1 in children's, Haemophilus influenza type B, Streptococcus pneumonia, and Achal also already mentioned the drama of the cost of a conjugate vaccine, about $130 per doses, and you need 3 doses to vaccinate 1 children. This is unsustainable, and the price will go up. Actually, I started working with Fila Institute in Cuba on their heptavalent conjugate vaccine. So we have to see always the limitation of this technology that we had no time. Of course, it's not a race. I'm better than you. This technology is better than that. But if we look forward, people in underdeveloping country or in rich country will die for pneumonia and for antimicrobial resistance. The 5+2 bacteria involved in all this, they're all capsulated bacteria, and mRNA vaccines are not the solution, also because, especially for the pediatric population, we have to admit that they are not the optimal formulation. So these are the limitations. This is where we are, and we have to think that there are other approaches. So where is innovation? So innovation maybe would be to find new conjugates approach to make new pneumococcal vaccines, lowering the price of what would be the $130 per doses of PCV vaccines. And an mRNA vaccine will not work on that. So it's glad to have everyone here, but we always have to go down, look how people will die and where are the limitations of each technology. So I'm just worried about the fact that the next dramatic death in children from pneumonia and for antimicrobial resistance cause number one of death today and from the future. We need to be smart enough to have technologies for everyone, and I do think the public institution will look at it in an honest manner, because $130 per one dose of a conjugate vaccine and $160 of the Moderna boost, sorry, is not the future for everyone.
Gary, you're waiting patiently.
Thank you, Andrew. Great comments, and Fabrizio, you nailed it. So I think innovation to me comes from also coming up with new models to get to low cost. You know, as my dad used to say, it's easy to drink good wine. What's hard is to drink good wine for cheap. Anybody can invent the best vaccine if resources are illimited, but resources are not illimited. And the model of funneling constantly public money, public taxpayer dollars into for-profit company to develop those products is of limited reach. It works really well. We saw it with COVID. We know this model works really well. What we know also is it comes with gaps. And that are the gaps that we are talking here to address. And again, we can develop all these great vaccines and all these mucosal immunity and name it. What we don't have yet is models that are bringing the cost down so that we are talking about access, universal access. So public funding that is going exclusively in a majority to for-profit for product that have no economic viability, like all these pathogens on this list, is a no. I don't know how this, why are people hoping this is going to work after 10, 15 years of seeing this not working very well? The example is the Ebola vaccine. It works super well, but it's not in Africa. Why is it not in Africa? Same for the monoclonal antibody. The model needs to be redeveloped to fill those gaps. Let the for-profit do their money. They are very good at it. They'll continue to develop great drugs, great vaccine for markets that are allowing that model to work. And for other model, by the way, there's great pharmaceutical company. Look at the Merck associate itself now more and more with IIV, which is a not-for-profit. I'm sorry, the Pfizer now unlocking their IP to 45 countries. We hope that this number is going to go up, including for cancer drug and all this. So the point is to bring that window bigger and bigger so that we keep the cost down of all these impressive technology, especially that they are supported at 99.9% by public funds from the start. Thank you.
Thank you. Can I ask a question on current challenges that we face? I mean, Sarah mentioned mucosal immunity and the need to develop vaccines that have that, but we also have a situation where, I mean, monoclonal antibodies that were registered are no longer, they've been stopped in the US because the Omicron variant's come along and it's undermined the efficacy. We've got vaccines that are increasingly struggling against the current variants. What is the model to, is it a pan-coronavirus? Is it to constantly update? What is the model for this?
Well, maybe we should give somebody else the chance to answer, but I mean, I think you can't have both very highly effective immunity and broadly protective immunity. You probably have to choose between the two. And there's a lot of work to be done in both of those areas.
Okay.
But combination, I'm sorry, combination therapy, I think, has a lot of promises as well. You know, most of those products went with one mAb, but combining them, of course, bring the cost up, but also improves the reach.
Achal has his hand up as well. And he's on mute. You've gone back to mute. Well done.
Thank you.
I think you've gone live.
Fabrizio raised this point somewhat sideways to your question. I think the issue with the pneumococcal conjugate vaccine illustrates perfectly one of the really important problems today. So we had 2 PCV pneumococcal conjugate vaccines from GSK and Pfizer available for the better part of about 15 years. But it took 15 years for the Serum Institute, because of the cost of development, while these 2 expensive vaccines existed, to create a low-cost PCV vaccine. And that, I think, illustrates in itself, the high cost of this vaccine, the 2 options that were available, in spite of Gavi's involvement, their relative unaffordability and inaccessibility around the world, and the time that it took to develop an equivalent to them, that was 15 years, which I think is kind of inexcusable when you look at what could have happened if those 2 vaccines could have been replicated more easily just when they came out, rather than have had to be completely reinvented from scratch, which is a process that took a lot of money and a whole lot of time, in which a whole lot of people didn't get the vaccine they needed.
Thank you. I see, Joe, you're typing answers to a couple of questions. Maybe since you've read those questions, could you speak and just paraphrase the question and say what your response to them in the question and answer box? It's a kind of couple of critiques, I believe, actually, so that you're responding to.
Yes, I mean, I was just looking at some of the questions around, patents, IP rights, whether the cost of those would, drive up any savings that the technology that the Quantum and others innovate. I haven't clicked send because my answer isn't very good, but hopefully you're putting me on the spot will help bring it out. And I think my answer to that is a little bit captured with what Fabrizio was saying. Not everything's going to work for every vaccine, but we have to, that every vaccine, every diagnostic needs at least a portal or a model that can make it at low cost. Because we know that we can simplify the technologies, and if we can do it into a container, like Quantum are doing with the WHO hub, that the process can be simplified. We cannot put the yellow fever egg platform in a container. That giant building on the left makes about 30 million yellow fever vaccines, and then the giant building on the right is well in excess of 300 million. Because the process is simpler, you can streamline it. And the cost is always much, much lower. So I think for these challenges around uncertainties, around patents and IP rights and things, I think we have the certainty we need to move. The Ebola example, Gary, you mentioned, is perfect. We have Ebola 12-hour drive away, I'm sure, from Dakar. It's in Guinea. There's reactivation cases that were sequenced in our labs last year. Same district where the Marburg case was first detected. Do we know where it is? So every healthcare worker there should be offered the choice to have such protection because it works so well. And certainly we feel that the vaccines for things like filoviruses should be transferred to the region so that we can be ready for it, because we would choose to make it at low cost. And then if it spreads beyond, then we can transfer it back out. But having a situation where people only make it when there's this hope, this perverse hope that there's a profit to be made, and then it doesn't materialize, and then the technology stops. That's what we need to address. And I hope that all together we can do that.
Brilliant. Thank you so much. Thanks, Joe. Els gets the last few minutes to wrap up and make some observations. And I just want to say my thank you to everybody, including Asia, who's in the room here, who's been organizing this for the last week or 2. So thanks very much, everybody. Over to Els.
Yeah, thank you, and indeed to thank you all, including Asia, for pulling this together and for your time. This has been a super rich conversation, but clearly we're only scratching the surface. I mean, each of the topics that we've touched upon requires more thinking, more discussion. But I think that what I take away from this is that amongst this group, at least, but it's a quite diverse group and very much involved in epidemic preparedness and response R&D. There is kind of a consensus on the importance of these platforms for vaccines, for manufacturing, for diagnostics, for treatments, etc. And this capacity to rapidly, I use the word adapt, but I'm happy to hear that many of you consider this like actually as innovation. It's not just adapting in the margins. This is the real innovation that will address the health needs, and that's what I'm very encouraged about, is all of you have also spoken about, like there are, the scientific and medical challenges, there are the technological ones around production, etc., and are around access and availability, and there is innovation needed in each of those, and that links to the business model. And I think we should move away from business model to a value proposition for health that needs to be sustainable, and where there can be business made by private sector actors, but it shouldn't be the goal. The goal should be to develop the health products, the health technologies that we need, and make them available where and when needed. And I would really encourage you all that as scientists that are so central, actually, where all that R&D and all that creativity and all that innovation emanates, that we all have to step up and engage in designing those value propositions and business models. We shouldn't be just a subject of an inequitable business environment or business ecosystem. We should be co-creating and demanding from our governments, from our leaders, from our funders, to do things differently, because it's the results of your collective work, of our collective work, that can actually get equitable access to people. And nobody stand in the way of that. We'll really use our creativity and our innovation to also, because some of it is technological innovation that makes equitable access more possible, or more available, or more rapidly, etc. And I think if we can make that case also to our political leaders, I think that will have a lot of impact, because now they think that, well, you're doing your work in the corner, and then there is a business model that will take it forward. It's not. We're part of the business model. We need to redesign it, and I'm very warmed by some of the businesses like José's and Petro's, your private companies, but that have actually embraced that goal of doing business for the public, for public health, for the public good. And it shows that it is possible to actually build something that is economically sustainable. And so I hope this can be a first conversation together that we can take forward. As you know, there is the Bellagio meeting where political discussions will try to pick up on what they've heard also at this meeting, and try to transform them into a political strategy to actually move this. It's not going to happen tomorrow, but bit by bit, and if we all work together and promote this narrative that you have so eloquently done, all of you today, I think we can get there. And so I want to thank you all, and also stay in touch. We will too, and try to put all the pieces of the puzzle together towards real transformative change. So with that, on behalf of all the organizers, thank you everybody, also all the participants online. We didn't have much chance to interact, but I'm sure there will be future opportunities. Thank you so much.
Thanks a lot. Bye. Thank you. Bye.
Thank you.